All right, g'day everybody. Welcome back to AI Australia. Nigel, how are you doing today?
I'm very happy to be in Melbourne. It's been a travelling month for me, and to get back here for a couple of days, meet some new people, super excited.
And last time we had the show, you were at Pause Fest. Good response to your talk?
It was, considering it was a pretty obscure topic on the subject of Australia's greatest startup and talking about genetics and gene tech and ag tech, and got an audience of about 250 and managed to make them laugh when on the subject of science, which is always a great thing and probably plays quite nicely into who we've got in the studio today.
It does, doesn't it? So I'll give a quick intro for our guest today, JJ Hastings. So JJ is an artist and a scientist, and her research explores self-experimentation, genome editing, machine learning, and the future intersections between tech and the human body. She has long-standing roots as a biohacker, having co-founded two community labs, London Biohackspace and Melbourne's Bioquisitive, and now has the first garage lab startup in Australia to be approved by the OGTR to work with genetically modified organisms. Her work has been exhibited at venues across Europe, India, Asia, North America and Australia. And JJ's career in scientific research spans over 15 years. She's an alumna of New York University, Harvard University, University of Oxford and Central St Martin's, with advanced degrees in biology, bioinformatics and fine art. And her research fuses and folds together the fields of machine learning, bioengineering, space exploration, new media art and ethics. Welcome to the show, JJ.
Thank you so much.
Wow.
Superstar bio. And any highlights from that period of time? Is it one of those things where everything's contributed a little to the recipe that is JJ?
I like to joke I know just enough to get into trouble in every one of those disciplines. Yeah, I've always been a bit of a provocateur as well as an outsider. I've never been, the ideal student. I've never been, I know that list sounds like it's really impressive, but I've never been, the A student in every class. Some of the classes, but not all. So I've never really considered myself an insider in any of the institutions or disciplines, but I certainly strive to be at least as competent or well-informed in every field that I've tried to work within.
How have you, does each field throw up a thread that you then start to pull and follow? Is that how you've gone?
Absolutely. In fact, now because of the work that I do, I'm falling deeply into law. So, so watch that space. There could be another degree after the PhD.
That's amazing. So just charting that course, kind of biology, bioinformatics, at some of the most prestigious universities in the world, really, really impressive. And you've had your art exhibited at venues all around the world as well, and you're at the forefront of biohacking. It's a pretty incredible story. Can you share kind of how it's unfolded and how you've got to this point?
Sure. So it was actually by accident that I ended up in biohacking. I was on track to end up in academia as a scientist. In fact, I've fallen back into it since becoming a biohacker unintentionally. But it was because I became horribly ill. So I was stuck at home going through treatment that required that I remain at home for a number of years. So I had to leave the ambition of an academic career behind, but the impulse was still there. So there was a room next to my bedroom that was a laundry room that was unused. So I managed to cobble things together and start creating my own ad hoc lab in that laboratory, doing very basic things. And this is when biohacking and DIY bio really wasn't a thing. Though, Nigel, as you mentioned, we've always been biohackers. So everyone tries to find a way to cobble together. Every researcher tries to find ways to cobble together under whatever bootstrap budget they've got. So I started looking for whatever materials, sources I could identify to get what I needed to continue doing the research that I found interesting. So becoming a biohacker was really just an impulse. It was an impulse that I couldn't ignore, even when I was feeling ill. So that turned into a new career as an artist, oddly enough, because I found an audience for the sort of work that I was doing and colleagues that sort of shared my interest in exploring biotechnology as a medium itself. And now you find me here doing my PhD in interdisciplinary research at University of Melbourne. I'm just about to finish that up. And so now moving forward with a career that is interdisciplinary in nature and a connection to biohacking that really is a reflection of my impulse for inquiry and experimentation.
So some labels there that are provocative on their own. Hacker, I think it does give a sense of fear into some people's minds, or perhaps not in the tech world. Have you chased being a provocateur? Is that always gone, I'm going to bust this open type of a perspective to it, or just pure curiosity that led you down holes that turn out to be quite provocative for modern society?
It's not always an intentional provocation. Sometimes I'm just really interested in something that makes other people feel rather uncomfortable. It's not something they don't want to think about, or haven't thought about really deeply, and so they're feeling a bit uncomfortable by whatever I'm unraveling in front of them. So back to biohacker as a term. Some have called it DIY bio to kind of soften it, I guess, and reflect more the education aspect of biohacking as an activity in a community. Some have called it community bio. Some have referred to it as being an independent scientist, and I guess that's really more what I'm an independent researcher. I utilize whatever tools I can find to follow whatever path of inquiry that interests me. So biohacking certainly has that provocative flair to it, because there is a public connotation that hacking is subversive, that it is meant to be counter to an existing system, right? That it is meant to pull apart and put back together, and therefore disrupts whatever power structure exists. So biohacking certainly has that connotation and makes a number of people rather uncomfortable. So, and I feel that discomfort myself. I'm not quite sure how I feel about being labeled a biohacker, though I am one, and being part of a community of biohackers. Some are quite overtly trying to subvert systems. So yeah, I think like I've said before publicly, I call myself more of a gray hat. I'm not intentionally a white hat or black hat. I just kind of pursue whatever line of inquiry is in front of me and that I'm interested in.
Yeah, you've clearly become comfortable with it, which is pretty awesome. I've just finished a book, Dark Emu, which is kind of an Australian book, and I've had my mind blown out a little bit. That hacks so many memes of Australian society and the hunter-gatherer myth of the Aboriginal people, the age of their society. They probably were the first bakers. They were mass agriculture, cities of 10,000 people, all of which have archaeologically been trampled into the ground. But the most interesting piece I found was the intentional development of plants and seeds and how quickly they were able to change their basic DNA to adapt them to being crops. Have you read the book by any chance? I
have. Unfortunately, I'm not all the way through it.
I've given away secrets. Spoiler alert.
It's fine. But I am forever intrigued by the deep history and knowledge of the traditional owners of this land. It is extraordinary, the knowledge that they hold and have held for so many thousands of generations. And so some of the stories that the settlers would relay back of wagon wheels pulling up rooted vegetables, thinking, oh my, they're so dense. Look what I've uncovered. You've gone through someone's patch.
The gods have provided for us.
Exactly. Yeah. Look how fertile this land is. Well, I wonder why it's so fertile. It's been cultivated in an intentional and remarkable way by a culture that's one of the most ancient on the earth.
And it's important. So many generations, the opportunity to actually modify the plants and the seeds in particular by their ability to pick particular seeds and recognize the qualities that seeds had for turning into meal versus replanting. It's highly scientific in its approach. And I think today I like Martian exploration as an idea. I'm influenced by people who are pretty major fans of that, but mainly for if we can survive on Mars, a much drier Earth or Australia, we can manage. I now realise we don't need to go to Mars. If we can just reach back 10,000 years into Australia's history, we know how to cultivate outer Victoria, where it's a desert. But goodness, getting that knowledge back out again is going to be a big challenge for us.
Oh, Nigel, do we have things to talk about? I'll have to come back for another podcast with what I'm doing starting this year.
Fantastic.
Yeah. And we've had a few guests on the show who've got connections with Melbourne University. So we had Tim Miller from Computer Science, we had Niels Wouters, obviously. Melbourne seems to be a great destination for the academic community these days. What is it that's so attractive about Melbourne, do you think?
What I found is that my colleagues here in Melbourne, and I teach at RMIT as well as a sessional, I teach design students and I teach materials research. So I would love to do much more cross-disciplinary teaching. So to open up my subjects, my studio practical lab-based subjects, to any student across the university, but particularly design and engineering. What I've found amongst my colleagues, both at University of Melbourne as well as RMIT, and even people that I've worked with in Swinburne, Monash, there is an openness here and a willingness to muck in. And I think also the isolation, just across Australia, sort of selects for extremophiles, which I think is why it attracted an extremophile like me. So as opposed to the sort of dense cultural cultivation of ideas in Europe and North America. Here in Australia, you're given a bit more fodder and a unique environment in isolation that I think has really bred some interesting ideas and collaborations.
And how long have you been in Melbourne for now?
This is now going on my third year in Melbourne, but I was in Perth before at University of Western Australia at Symbiotica, which is also another interesting organization, research group that has been around for over 20 years now, that intentionally forms unique collaboration and disruption across different disciplines within an institution in Perth.
We were talking about, obviously our show's a big focus on AI, and looking through your background, and you talk about kind of biohacking, CRISPR and AI in the same kind of sentence. And when I first started thinking about this, I kind of saw the biohacking and CRISPR over here and AI over here, kind of pursuing slightly different goals and objectives. Clearly that's not the case. How do you see these things coming together?
Machine learning, and I think I'll expand it out to machine learning, not just AI, because I think that tends to narrow the definition of what we're talking about. AI is a pursuit for a particular kind of application sometimes, depending on who you're talking to. In my mind, machine learning, something that, oddly enough, I've been working with since my bioinformatics days at Oxford, when we started talking about neural nets and support vector and Markov models, it was really, to me, not really, it was a great statistical model, a great way to analyze large data sets, but really wasn't applied in a stochastic way. Now it's moving into stochastic and even decision-making models. And that to me is interesting because now we're talking about designing life. So we're moving away from simply understanding large data sets towards the formation of decision models. And from that comes a whole new avenue for research and inquiry in science, particularly when it comes to the body. What does it mean to have completely digitally designed, completely machine designed life? What does it mean for, in the case of the Human Genome Project, and please correct me if I'm wrong, my recollection of the Human Genome Project is that it's the human consensus is really more an average of 25 individual genomes. So the model that we're using of the human genome, we're now getting into resolution where, like I had my whole genome sequenced in 2016. I now have the complete sequence, all 6 billion letters of my genome sequenced. But even that is really a sort of average of the genome that's within each cell in my body. It's not really representative of the complexity and uniqueness of each individual molecule, each individual chromosome that one can find. I feel like I'm going off on a tangent here, but
I'm starting to understand why something as complex as artificial intelligence and machine learning is required, though, because you're describing biological models in mathematical terms.
Yeah. Yes, well, and we're converting biological, chemical information into digital, into binary. I'm interested to see how this shifts as we move into quantum computing. But for now, we're relegated to binary to describe a quaternary and now even octenary with new synthetic nucleotides. What that now means for completely artificial life.
Yeah. There's a strange echo, who said it? History doesn't repeat, but it echoes of a century ago, the turn of the 1900s. We start seeing quantum physics becoming mainstream and replacing Newtonian physics, and it's just probabilities and maths to describe particles in a way that now you're describing biological things, which we think of as fleshy and organic, also. And perhaps that makes sense because I suppose squishy and organic things are made up of atoms as well.
Well, I saw the description the other day of, we're shifting to an age of theoretical biology now, where we were moved for theoretical physics a long time ago. But now with data and machine learning and what we understand about the genome, we can now start to do theoretical biology, which was really interesting.
Yeah, well, and now imagine I like that analogy, actually, of quantum physics. And also the sort of, I wouldn't say threat. I think threat is too strong of a term, because unlike nuclear physics, I can't quite destroy a city. I think that's highly unlikely. And just to sort of tangent back to being a biohacker, there is palpable and often very vocal concern over the threat of biohackers, that they could pose a real threat in terms of bioterrorism, or creating I guess the most common trope I've heard is being able to synthesize or create some sort of virus in their garage lab. And the response I've had for the past 10 years is it is more likely they will give themselves really horrible gastritis by transforming or cultivating a particular bacteria than being able to actually put together something that costs thousands of dollars and will likely hurt them sooner than it hurts anyone else. Not only that, but the argument I've heard from very competent people and people in the know is that it's really those who are in institutions that have access to the materials that are of greater risk than biohackers. So biohackers as a risk, I will say it's highly unlikely.
Right. I'm comforted by that, James. I reckon it's more likely there'll be some nasty virus on my laptop than there will be something honest, genetically modified.
Yeah, no, my experience mostly is they're more likely to produce flack from regulatory bodies and the media than they are to actually be a real risk to other humans.
And I've hugely enjoyed a bit of research ahead of this podcast. I've really found my groove over the last few nights, and I haven't been sleeping much because I've been reading a lot. But I've just been amazed by how far along some of the technology in this space is. And one of the examples I saw was a company I believe called Oxitec in London, who have been able to genetically edit mosquitoes to combat dengue fever, yellow fever, Zika virus. Yes. And how they described it was that they release male-only mosquitoes, because I didn't know this, but male mosquitoes don't bite. It's females that bite. They're genetically modified. They mate with the females, and the offspring have effectively a ticking time bomb in them where they just die. Mm-hmm. And where this has been trialled, I think in Brazil and a few other locations, they've wiped out the population of mosquitoes by about 90%. And the state of Florida in November voted to trial this in Florida to combat the spread of the Zika virus. Mm-hmm. And on one hand, you think mosquitoes cause carriers of death for generations, we can deal with that. And on the other hand, you think, what are the unintended consequences of removing a species from our ecosystems?
Yeah.
It just amazed me that we were at that point already.
Yeah. Well, look, I'll say gene drives, as opposed to other modes of culling a species, simply seem a bit more extreme in that they're not quite directly under our control. There are badger culls, there are calls to exterminate rabbits or frogs or what have you, but it's a more direct, controlled way of carrying out a policy. Gene drive, or what you're describing of modifying a population or to wipe out a population that are carriers of a particular disease, I think it makes people uneasy because it's out of control. It might have knock-on effects that we aren't able to model as yet. There are a number of studies, though, in a number of labs across the world that are doing intentional release within a contained environment to try and model this before release into the environment. So I'm not entirely sure that the use of gene drive is going to be widely used, but I see it really as an extension of what we already do in terms of population control, and in some cases could be really useful, not just for human health, but also for ecological needs as well.
So because I come from a household of artists, not scientists, James, I'm more interested in your work around the art and And it seems to me like in some ways you're pretty comfortable with not knowing how this is going to end when you start a project. If I had to summarise some of the descriptions of your art, that would be it. It was like, just let's do this and see how this emerges accordingly. Do you want to explain for the audience what was the art you were doing? Explain the work a little bit.
Let's see. I've got a few different projects. So I think you're hinting at the Demiurge.
Yes.
Is that the one? Yeah. So the Demiurge is turning into a wider project. It started with a commissioned work for the Science Gallery for their Perfection exhibition. And it was an idea that I'd been knocking around and playing around with on my own, and then they gave me the support to scale it up for the exhibition. The Demiurge took my genome, my whole genome sequence, and would then give me instructions based on its decision tree of what I should modify in my genome to make it more perfect. But that includes instructions to give to CRISPR. So it gave me the sequence that I would need to give to the enzyme to edit my genome. And then I could take that construct that I've built and change my body using that, my somatic cells, using that CRISPR sequence.
And you would just unleash that as a work of art and see what happened next. But I think that's kind of the scientific pioneering spirit, to be honest. I mean, there are so many genetic experiments, there has been no idea whether it was crossbreeding or, in the most simple sense, plants, grafting, been a million ways in which our species have intervened in biological things over our civilization. But yes, I think art's a great way of broaching the subject with a community of people who are not going to engage in reading about CRISPR. Now, is it true? Like all I know about CRISPR is a Chinese scientist who's been disgraced. Is that pop culture, or what's the story behind the scientist who has used it?
It's still unfolding. There is still a lot that we don't really know because this is a project that's been going on for years in the making, with multiple intersections internationally and a lot of private money and other players that we still have not quite uncovered yet. I think I'll state my position in that what he did, the act of editing children, of interfering with their genome, is not entirely unjustified. There are many reasons why genetically engineering of editing out diseases that we know only require one gene are highly, possibly lethal, or could affect their overall well-being. There is justification for genetic engineering of modifying our bodies and even selecting embryos. What Dr. Ho did was unfortunately something that led to a blowback in the global community because it was not open to oversight, because it was done to a target, CCR5, that really does not have much justification for editing other than making the children resistant. The children, the twins that were born, are not completely immune to HIV. The CCR5 allele makes them more resistant to it, but not completely immune. There is now speculation that it was done to augment their cognitive function. This connection between CCR5 and cognitive function was only studied in a small cohort of mice, and one study, as far as I'm aware of. So it's an unsubstantiated claim. So the target itself is what I think most of the community, scientific community, ethical community worldwide is concerned with, that it was done in secret without any sort of openness, transparency, or oversight, that it was done by private investors, by an individual that really does not have a track record in using this technology, let alone to treat and intervene with human patients. He was working in other animal models beforehand, and before that in biophysics. So there are many things that are problematic from an ethical standpoint, whether or not the work was justified, the risk of whatever he was doing was justified, whether it was right to do this and the impact that it would have on the global community that is trying to do very serious research into genome editing to eradicate deadly disease. So I think the concern with people like her who are working outside of institutions is that they're going to make leaps beyond their capabilities and therefore do more harm than good.
Right. And so compared to Dolly the sheep, which was how long now? That the 90s?
Yeah, I believe, what was that, 2000? No, it was 96, I think.
Yeah.
Yeah, 96.
So open science, cloning an animal for genetic gain, I guess. I actually lost the context for Dolly the sheep. But
It was cloning. So they were doing somatic nuclear cell transfer. So it was the test of whether or not one could clone an animal.
So no real intervention in any kind of DNA of any kind.
It's just taking the nucleus out of an adult sheep from her mammary gland and putting that into an oocyte.
Right.
So it was actually taking a single adult organism and trying to reboot the nucleus into a whole new animal.
It's funny how we bring our own ethics to the table in some ways, having been a family who went through IVF for so long, endured so many tests and kind of the nuchal fold tests and all the moral and ethical decisions you'd have to make. I've just got comfortable if somebody had said to me, well, why don't we, your grandmum's got Alzheimer's and diabetes, do you want us to modify that? I'd be ticking that box straight up. But that's my personal ethics. How's it translating into the law? Is the law 100 years behind on these things or 10 years behind?
This is something I'm trying to figure out because I'm not already a legal expert. I'm working with experts in law, but even they're struggling because there aren't enough people that are competent in both. And I think this is where I try and work, where I have enough to be dangerous in both fields, to know where the problematic areas are and try and not necessarily find resolution, but at least prompt the discourse that needs to happen to figure out the way forward. I'll step back a little bit to IVF because there's an interesting aspect to this. When we're talking about AI, when we're talking about machine learning, what we're talking about is a statistical model or a very deep analysis of human data, of human-generated data, right? So machines are only ever as strong as the data they're given. And they're biased. We talk about bias a lot, but they're biased by whatever information we have given them and the way that we've classified it for them. So they can only ever learn what we have given them. So really, AI, I'd like to say, is AI is the most extraordinary circus mirror of who we are as humans. It's a distorted view because it's amplified, but it's really just showing us precisely who we are in a different manner. When it comes to humans, if you think about the application of AI towards Facebook and other social media, the way that it's shifting the information that humans act upon as well, including our dating lives. So you want to talk about IVF, that's a very direct way of intervening with human reproduction. Facebook is shifting who we date and pair up with. And dating applications have been selecting for us who we should be paired with. So machines are already shifting human reproduction in a more subtle way. And so they're forming their own sort of ecosystem around human evolution and the decisions that we make, not only for our own health and well-being, but who we reproduce with, who we pair up with, and who we consider a reliable source and want to be around. So I think AI is fascinating, not necessarily because of the technology or the way it can do rapid analysis of the questions that we might have, but rather the way it reveals back to us our starting biases, the need for better training sets, the need for, when it comes to health particularly, when we talk about what diseases we want to cure and edit out of ourselves, part of the purpose of the demiurge was to also unpack the way that we treat disease. What does it mean for me to have a mutation in my genome that could also be responsible for making my mind the way it is, or my personality? What is worth editing in ourselves and what is worth keeping as a possible error from our own position of health and well-being?
So you printed your full sequence.
Yes.
Worried about that intellectual property, getting me just snapping it on the wall at an exhibition and the things I could do with that. And I'm sitting here as someone who was just desperately curious about his DNA in terms of migration and sold my minor sequences to the Mormon Church and Ancestry.com. What's your take on that in terms of is it a security issue, the data associated with our biology?
Look, the sequence itself is neutral, and we only differ by a small fraction, a fraction of a fraction. So what differs us, that point of distinction, can be powerful. I'm not so worried about what you might do with my genome sequence. And I did purposely obfuscate. I did purposely obfuscate some of that information in the final exhibition because there are legal concerns over privacy, because it's not necessarily relevant to put my whole genome sequence. And to be honest, most people really wouldn't be able to do much with that information. Not because if someone wasn't highly motivated, they couldn't, but rather the payoff is rather low. I'm not terribly concerned about that. But what I am concerned about is, so I've taken my whole genome sequence and I keep it on a secure encrypted disk, right? It's not uploaded anywhere. It's kept on a private hard drive that is encrypted. So there are segments of my genome that are available through like 23andMe or other databases. And those, I think I have more concern over privacy around 23andMe reselling my information. So taking my genomic data, aggregating it with others, and reselling it onto other clients. So appropriating my information for their own financial gain without my consent. The second is forensic use of that data. We need to come up with some way of securing access to our genomic information. Right now, it just takes a warrant, and you can always have a knock-on discovery in that database. You can basically do the same thing you do with any other gene that you sequence. You can throw it into a database and see what hit you get. And that's not really going to be effective because that will lead to surveillance that is unwarranted.
And this got a bit of interest in I think I read the case in California there was a horrendous serial killer in the 70s and 80s, I think, the Golden State Killer, who went uncaught for decades, string of horrendous violent crimes, who was actually caught through a DNA sample that he provided to 23andMe or Ancestry or...
I think it was another platform, but yes, essentially, he wasn't the target. They weren't looking for him. It was a family member. So he was a knock-on discovery that cracked a decades-old case that was horrific. But it also makes us question: at what point do we need to limit that access or ability to use that information to actually bring someone to justice?
And one of those organisations, I think, just kind of announced in November that they were changing their T's and C's to allow FBI access to that data, and that just applied to everyone and everyone who provided DNA even prior to them changing the T's and C's, and it's now accessible to the FBI. And I've had a few conversations about this, and people have said, well, they've caught one of the most horrific serial killers in American history. That's a great outcome.
Which is marvelous, until they're suddenly searching for people that have done misdemeanors.
Yeah. Yes, well, as Australians, James, the AAB probably doesn't give us any cause to be too cocky about the state of the moral and legal environment in another country. So,
it probably doesn't, does it?
No. We've wandered into science fiction here, and I'm just wondering a little. Are you a fan of science fiction?
Yeah, from the time I was a kid, I grew up on Star Trek with my dad, who was not really into sci-fi, but certainly stoked my interest and imagination. Some girls liked to play with dolls when they were younger. I had a great doll collection that my mother bestowed on me, but I preferred to go outside and run in the forest and get muddy and play with lizards. And then when I was younger, I used to memorize the Mercury and Apollo and Gemini programs and fascinated and fixated on everything to do with space. So I was a huge nerd.
You're among friends here, yes. I can tell you that.
Definitely a sci-fi fan. Ursula Le Guin, Ray Bradbury, people that tend to fixate not just on the technology, but its deep implications, the way it shifts and shapes who we are as a species.
I always describe sci-fi, and I'm a bit of a fan as sort of psychological thrillers set in another time or place.
Yes. Yeah.
And it's all about those tensions and underlying things. So someone's given me Change Agent by Daniel Suarez as a book, which is a CRISPR crime story of some description. So I'm looking forward to that one. What about movies? We're always debating, what's the movie that depicts the most likely future that's playing out today? And this sort of started with Toby, really, with writing 2062, his book about the world that AI made. 2062 is the year that Jetsons is set in, just for those who need a bit of pub trivia. We almost need to change the range with someone who's a scientist of your, because we're in Gattaca, altered carbon.
We should probably have Minority Report.
Minority Report.
We've got Blade Runner. We've got Star Trek.
You know what? The Island. That I wouldn't say it's the best movie I've ever seen, but the story and concept is compelling. Ewan McGregor and Scarlett Johansson, I think. Yeah.
Well, please explain the plot.
So it turns out that we humans are fully cloning ourselves, and they are kept in, quote, the island. And that island is inhabited by clones that think they're living in this ideal Truman-esque world, and they win the lottery. And when your lottery your lottery number is pulled up, you are given a special trip off the island. And in fact, instead, what is unraveled is this trip is a surgery that then culls them for their organs, for their real avatars in the real world. So it's certainly horrible to think of your clone, of your twin, an alternate world purely for your benefit. But it is the story of these clones discovering that they are, in fact, held at the behest of their twins outside.
These incredibly wealthy individuals who obviously can afford to buy the clone, and it's kind of brand new, fresh off the production line, and they upgrade their body with the clone from the island. It's a fascinating story. You're right, the film's not great, but the concept is pretty cool.
Certainly it's stuck in the back of my mind quite deeply.
I think that form of the art, that's not your art. Are you a filmmaker as well, by any chance? I would not be surprised.
I like making small speculative films around my work as a way to sort of unpack how I think they might play out. But of course, that's speculation. I have no idea. And that's part of why I do the provocation. I'm interested in the public's reaction, in the reaction of scientists that are also working with the same technology. I'm interested in how it intersects with a number of different ideas and how it might play out. My work isn't necessarily intended to be the direct solution. I'm not posing a solution by any means. I'm showing a possibility and letting that possibility unfold in the public sphere.
Really trying to stimulate thinking or open people's horizons, broaden people's horizons a little?
Yeah, one is raising public awareness, right? So most of the public, as you mentioned, Nigel, aren't in direct contact with people that are working at the avant-garde of AI, of synthetic biology, of genome engineering, and even medicine at the highest levels. My work gives them an opportunity to see one version of it, usually a sort of hacked together, lower level version of the tech, but at least see how it can manifest into something that will have a direct impact in the near future. And so I'm interested to see what the public thinks, but also reflecting back to scientists that are working in their own spheres, how the intersection might play out. What does it mean for someone that's working in AI to suddenly apply it towards designing biology? Not just, bacteria as we might with synthetic biology, or synthesizing a full yeast genome as we're doing with GP Wright, but rather, what does it mean then to fully design life? Fully digitally, fully machine-driven, and then refabricated into the carbon-based life that we exist in.
And that concept in programming life, and this is something that I've, as I said, reading the last few days and realizing that I'm quite new to this topic. And I kind of felt like if I was new to AI and I spent a weekend reading about AI, I would probably reach the conclusion that artificial general intelligence was weeks away, and because of the media hype. I'm kind of interested now, I'm going to throw myself into a different space. Is my perception that we're very, very far advanced in programming life? Is that similar kind of hype to artificial general intelligence, or are we really quite close?
Well, AGI is the endeavor to try and reproduce human thought. It starts with this sort of hubris that we can, in some way, that we actually
We even
understand
understand
what we
it
are.
is, yeah.
Right, exactly. That we can understand both the aggregate of our conscious and unconscious and distributed notions of intelligence within our bodies, let alone socially, how it would interact with another AGI. So it starts with that hubris that you can reproduce it. You can reproduce some sort of simulacra or another version of it in some way that is interesting. I think AGI research is valuable in unpacking and teaching ourselves what it means to have intelligence and creativity. But yeah, when it comes to synthetic biology, when it comes to genome engineering, we're making huge advancements. And I guess I'm concerned in certain parts of the world that don't have direct access to the technology like we have in the States. In the US, having CRISPR available in your local community lab is a given. It's not even having access to synthetic biology is what kids do in their science projects. In Australia, in Europe, in parts of Asia, certainly the African continent, access to the technology is removed. It's something that's kept within specific spaces and only provided to certain people. So this will lead to a gap of understanding. And this is a concern of mine, particularly as we try and make these grand leaps forward. Imagine if AI wasn't at all accessible, if machine learning was something that you had to have a budget of $10,000, $20,000 to do. So it doesn't cost that much for synthetic biology, but in certain parts of the world, it is a huge barrier still. We are now working towards fully digitally designed and complete synthetic genomes. I think we still have a long ways to go because, like the endeavor for AGI, we still don't quite understand the genome sufficiently. What are we printing? What are we synthesizing? We're synthesizing the aggregate or some sort of bricolage of genes and non-coding sequences. What we have to ask ourselves, do we fully understand what we're printing? And will it function in the same way as an original individual single molecule genome? We still don't know. And I think there's an interesting parallel there to the pursuit of AGI.
With CRISPR and that technology, and we talked about accessibility and cost, and as I started researching, the first thing I see is an ad where I can buy a CRISPR kit for $300.
Yes. And I'm swatting down those stories left and right from press.
That's kind of, is this some kind of scam, or is this real? I mean, what can people do?
Well, I don't want to get into
$300?
too much trouble here because I do know the person that is selling these kits. I've vocalized directly to him my concerns multiple times, and I have purchased one of these kits myself. I have purchased one of these kits myself and tried to use it within a sanctioned space, and it didn't work. It didn't work. In fact, it's been reported in multiple places that the kit is either not containing what it says it's containing, doesn't function as claimed, or possibly contains other things that unintentionally are present, certain pathogenic strains of bacteria. So there's quality assurance issues with those kits. Yes, they're cheap, and I would fully endorse. In fact, I would love to. I've talked directly with the leadership of regulatory bodies here. I'm talking with experts here in Australia to try and create our own Aussie-based kit that would be accessible, as well as providing an infrastructure of guidance around them so that it can be possible for students and startups and anyone else to be able to work with the technology, because that is a gap that we need to address here in Australia and New Zealand.
With that kit, what kind of things would, with a well-designed kit where quality is taken care of to a certain standard, what are the kind of things that people could do with that? Well,
ideally, you'd only be doing a limited number of things to learn how to utilize the technology and modify it. So just as you would give someone, a basic kit of how to pull apart a basic computer and build one, given certain components, like a Raspberry Pi type kit, right? Mm-hmm. Arduino Raspberry Pi. You give them the basics of physical computing, plus a whole menu of different components they can play with, and then you let them play with it and figure it out. Then if they want to pursue something more sophisticated, so something out of established models that use like a simple lab strain of E. coli that is totally tested. And I know E. coli, has an association with gastritis, but E. coli is everywhere in your body. It is pervasive. And to put it in really crass terms, you poop out more E. coli for bowel movement.
There's probably more E. coli than humans, that's for sure.
Exactly.
Might be their planet we're on, James.
Exactly. So E. coli, I know when you say you have E. coli in the kit, that sounds really scary. And if it gets transfected with the wrong thing, it could possibly give you a nasty, stomach bug. But it's so well characterized and used so often in the lab, and it even if it gets out, it is unlikely to live because they're really heavily cultivated. So the risk to the environment and to the individual is very low with these established strains. If they wanted to work in more sophisticated models, they could then go through a process of review and be given access to more sophisticated tools.
It might just create one more scientist, James.
One more is always good.
That's one more is all it ever takes, is one more curious scientist, and we end up here with the most remarkable exploration of the territory of three fields I've never really known much about. Bioinformatics, in summary, what is that space precisely?
It's very simply the application of mathematics and computation to biological data sets. So with the emergence of massive sequencing initiatives, of data sets around structure of proteins, so with this aggregate starting at the turn of the millennium, we had this massive aggregation of data, and we really didn't know how to analyze it. So in came bioinformatics to try and call through all of that, to try and understand this massive wave of information that was coming at us. And it's now becoming much more sophisticated, so applying deep learning and other forms of AI towards those problems so that we can, with now much more sophisticated, much more expanded data sets, try and get to grips with what we're studying and sequencing and discovering. We still know so little about biological life on this planet, let alone other planets. We're trying to get to grips with it. And bioinformatics is one approach to trying to understand all of that.
Modelling biological data at scale and
Yes.
make predictions based on that data and try to understand or predict what's going to happen.
Exactly.
Well, James, that's been the most fascinating 55 minutes of my life. That is I've learned so much. I think, you're five amazing people in one. So if your DNA of science spreads out, there will be so many jobs, because you'll need a lawyer, an ethicist, a physicist, a mathematician, a biologist, all in the same lab working together, and that's a pretty exciting future.
It's amazing. There's one last question I do want to ask, though, really quickly. BioQuisitive, your lab here, I'd love to hear a little bit more about Bioquisitive is all about, and if people are interested in this space, and if people in our audience are interested in this space, how can they kind of find out more and get involved?
Absolutely. Bioquisitive is part of a global network of community labs. So some have called them biohackspaces. I think community lab is a better descriptor of it. So Bioquisitive as a community lab is a place where anyone can rock up and check out how a lab works, because it is a functioning lab, OGTR approved, PC1 established, so you can do genetically modified GM dealings in the lab. You can learn how to do basic microscopy, how to do basic cell culture, how to work with plants, how to design your own plasmid for use in GM. So it gives the community a space, no matter where you are in your understanding or background in science, no matter what your interest in science, you can come to the space and form interesting discussion and collaboration within Bioquisitive.
And it's the first lab of its kind in Australia, is that right?
No, that's not. So BioFoundry in Sydney was the very first. BioBase is now showing up in Brisbane. And there's another group that's not really official with their own lab, but there is an active group in Perth. So there is definitely some interest in Australia in biohacking, DIY bio. But Bioquisitive is our local group here in Melbourne.
All right. And the idea is just getting people more engaged in this topic, getting people, educating and access and skills.
Yeah. So there is skills acquisition to this, but it's also just a space to actually talk about science. You can have some water cooler discussion that's really interesting, but you can be around some other people. We have established researchers, we have homeschooled kids, we have people with all sorts of different political views, all rocking up and having incredible discussion and actually working together, trying out the technology directly.
And is it a contentious issue in Australia? Or is there strong kind of vocal groups who are against the idea of GM in general, or is it pretty harmonious? Or
we do have some groups that are rather active and have already decided their opinion of what GM means for Australia. We thankfully, we don't want to avoid those difficult conversations because that doesn't lead anywhere, right? But we do want to create a protected space where we can at least try it out and come to that difficult conversation together and have direct experience and access to the technology in question so that we're informed, we're capable, and are on somewhat equal footing as we talk about the future of the technology itself.
Awesome. JJ, it's been amazing talking to you, and thank you so much for being so generous with your time, and best of luck with your future ventures. I'm sure we'll be talking to you again at some point when you've added your next one of many disciplines to your bow.
It would be a pleasure. Thank you for having me.